Alright, let's talk about something that might not get the same spotlight as the heart or the brain, but is absolutely critical to how our bodies work. I'm talking about glycosaminoglycans, or GAGs, proteoglycans and glycoproteins. These aren't just random long names you have to memorize for your biochemistry exam. They are the unsung heroes working behind the scenes. Think of them as the body's architects, engineers, and communication specialists, all rolled into one. They build our tissues, lubricate our joints, and even help our cells talk to each other. Understanding them is not just about passing a test. It's about grasping the fundamental principles of human health and disease. These molecules are everywhere, from the cartilage that cushions your knees to the mucus that protects your airways. They form the extracellular matrix, which is basically the scaffolding that holds our cells together, giving tissues their shape and strength. When you think about it, without this intricate network, we'd be little more than puddles of cells. So when these molecules aren't made correctly, or if the body can't break them down properly, the consequences can be devastating. This leads to a group of serious genetic disorders that can affect everything from skeletal development to cognitive function. That's why we, as future doctors, need to know this stuff inside and out. So why should you, a busy medical student, care so much about these complex carbohydrates and proteins? Because patients will come to you with symptoms, not textbook diagrams. A child with skeletal abnormalities and cloudy corneas might have a lysosomal storage disease related to these molecules. An adult with joint pain might have an issue with the cartilage that's rich in proteoglycans. Knowing the underlying biochemistry will help you connect the dots, make a diagnosis, and understand the treatment options. It's about translating that complex science into real-world clinical practice. It's about being a better doctor. In this guide, we're going to break it all down. We'll walk through how these molecules are built, what they do, and what happens when things go wrong. We'll cover the high-yield facts you absolutely need for your board exams, but we'll do it in a way that makes sense, so it actually sticks. We'll talk about diseases like the mucopolysaccharidosis and eye cell disease, and I'll give you some simple ways to remember the key differences. So, grab your coffee, get comfortable, and let's dive into the fascinating world of our body's microscopic architects. This is knowledge that will serve you well throughout your entire medical career. Let's get into the nitty-gritty of how these molecules are actually made. Think of it like a sophisticated cellular factory with a very precise assembly line. First up, we have the glycosaminoglycans, or GAGs. These are long chains of repeating sugar units. The process starts in the cytoplasm, where simple sugars are converted into activated forms like UDP sugars. These are the basic building blocks. Then the real action happens in the Golgi apparatus. Here, enzymes called glycosyltransferases add these sugars one by one to a core protein, creating the long GAG chain. An important detail is that most GAGs are sulfated, which gives them a strong negative charge. This happens after the chain is built, also in the Golgi. Now, what about proteoglycans? It's actually a pretty logical next step. A proteoglycan is essentially a core protein with one or more GAG chains attached. So, the cell first makes the core protein on the rough endoplasmic reticulum, just like any other protein. From the ER, this protein travels to the Golgi. In the Golgi, the assembly line for GAGs kicks in, attaching those long, negatively charged chains to the core protein. Imagine a bottle brush. The wire is the core protein, and the bristles are the GAGs. This structure is key to its function, which we'll get to later. The type of core protein and the specific GAGs attached determine the final proteoglycan and its job in the body. Glycoproteins are a bit different. While proteoglycans are mostly carbohydrate by weight, glycoproteins are mostly protein. They have shorter, more branched carbohydrate chains called oligosaccharides. The synthesis here can happen in two main ways, which we'll compare later: N-linked and O-linked. For N-linked glycoproteins, a pre-made block of sugars is built on a lipid carrier called dolicol phosphate in the ER membrane. This whole block is then transferred to an asparagine residue on the growing protein. For O-linked glycoproteins, the process is simpler. Sugars are added one at a time to a serine or threonine residue, usually in the Golgi. So, to recap the factory analogy for all these molecules, the protein parts are made in the rough ER. The complex carbohydrate additions and modifications happen primarily in the Golgi apparatus. This ER to Golgi pathway is a central concept in cell biology, and it's absolutely essential for producing these molecules correctly. Any defect in this pathway, whether it's a missing enzyme or a problem with transport, can lead to serious trouble. It's a beautifully orchestrated process, but it's also a vulnerable one. Understanding this assembly line is the first step to understanding the diseases that arise when it breaks down. So, we've built these complex molecules. Now, what do they actually do? Let's start with proteoglycans. Remember that bottle brush structure with all the negatively charged GAGs? Those negative charges attract a lot of sodium ions, and where sodium goes, water follows. This means proteoglycans act like giant sponges, soaking up water and creating a hydrated, gel-like substance. This is the foundation of the extracellular matrix. This gel resists compression, which is why it's so important in cartilage. It provides the cushioning in your joints, allowing them to absorb shock every time you take a step. Without it, your bones would be grinding against each other. GAGs themselves, even when not part of a proteoglycan, have crucial roles. Hyaluronic acid, a unique non-sulfated GAG, is a fantastic lubricant. You'll find it in your synovial fluid, which keeps your joints moving smoothly, and in the vitreous humor of your eye. Other GAGs, like heparin, are powerful anticoagulants. Heparin is used clinically to prevent blood clots. It works by activating a protein called antithrombin 3, which then shuts down the enzymes that cause clotting. Heparin sulfate, a related GAG found on cell surfaces, plays a role in cell signaling and adhesion, acting as a co-receptor for growth factors. Glycoproteins have an incredibly diverse set of jobs, largely because the protein part can be anything from an enzyme to a hormone to a receptor. Their main role often involves recognition and communication. The carbohydrate chains act like little flags or ID badges on the cell surface. This is how your immune system recognizes your own cells versus foreign invaders like bacteria. They are also critical for cell-to-cell adhesion, helping to form tissues. Many hormones, like TSH and HCG, are glycoproteins. The carbohydrate portions help with their stability in the bloodstream and their ability to bind to their specific receptors. Think about the big picture here. These molecules are the glue, the lubricant, and the communication network of our bodies. Proteoglycans provide structural integrity and cushioning. GAGs provide lubrication and regulate processes like blood clotting. Glycoproteins are key for cell identity, signaling, and forming stable functional proteins like antibodies and hormones. They are all working together to maintain homeostasis. When you see a patient with symptoms like joint stiffness, easy bruising, or recurrent infections, these are the kinds of fundamental processes that might be going wrong. It's a testament to how biochemistry directly impacts clinical presentation. A system this important needs tight control. The body can't just be making these molecules non-stop, nor can it let them build up forever. The regulation of synthesis is complex and often tied to the needs of the cell. For example, the production of proteoglycans for cartilage can be stimulated by mechanical stress and certain growth factors like TGF-beta. This makes sense. If a joint is being used more, it needs more cushioning. The synthesis is controlled at the level of gene expression for the core proteins and the many enzymes involved in building the GAG chains. It's a highly responsive system that adapts to the body's changing demands. On the other side of the coin, we have breakdown, or catabolism. This is just as important as synthesis. For proteoglycans and GAGs, this job falls to a special organelle inside the cell, the lysosome. The lysosome is the cell's recycling center. It's filled with powerful digestive enzymes called acid hydrolases that work best in the acidic environment of the lysosome. When a proteoglycan gets old or damaged, the cell takes it in through a process called endocytosis. Inside the lysosome, a whole team of specific enzymes works in a stepwise fashion to chop up the JAG chains and the core protein into their basic building blocks, which can then be reused by the cell. This breakdown process has to be perfect. Each specific bond in a GAG chain requires a specific lysosomal enzyme to break it. If even one of these enzymes is missing or defective due to a genetic mutation, the breakdown process stalls. The GAG chain can't be fully dismantled and it starts to accumulate inside the lysosomes. This is the fundamental problem in a group of devastating genetic disorders called the mucopolysaccharidoses, or MPS. The lysosomes become swollen with undigested material, which disrupts normal cell function and leads to the progressive symptoms we see in these diseases. So, the balance is key. Synthesis is regulated by cellular signals and growth factors, building what the body needs when it needs it. Breakdown is handled by the lysosomes, cleaning up and recycling old molecules. This constant turnover ensures that our tissues remain healthy and functional. The regulation of glycoprotein breakdown is similar, also involving lysosomal enzymes. When this delicate balance of synthesis and degradation is disrupted, especially on the degradation side, the cellular consequences are severe. This is a core concept for understanding a whole class of inherited metabolic diseases. Now let's talk about what happens when that lysosomal recycling center breaks down. This is where we get into the mucopolysaccharidosis, or MPS disorders. These are a group of inherited lysosomal storage diseases, each caused by a deficiency of a specific enzyme needed to break down GAGs. Because GAGs are found in so many tissues, these diseases are systemic, affecting the skeleton, heart, liver, spleen, and often the brain. The undigested GAGs build up in the lysosomes, causing cells to become engorged and dysfunctional. This leads to progressive damage throughout the body. There are several types of MPS, and for your exams, you need to know the big ones. MPS 1, or Hurler syndrome, is the most severe form. It's caused by a deficiency of alpha-L-iduronidase. Patients have coarse facial features, corneal clouding, skeletal deformities, what we call dysostosis multiplex, and developmental delay. MPS 2, or Hunter syndrome, is unique because it's X-linked recessive, while most others are autosomal recessive. A good way to remember this is hunters see clearly, no corneal clouding, and aim for the X. They have similar symptoms to Hurler, but typically no corneal clouding and a different pattern of aggression. Other types include MPS 3, Sanfilippo syndrome, which primarily affects the central nervous system, leading to severe behavioral problems and dementia. MPS 4, Morquio syndrome, is characterized by severe skeletal dysplasia but normal intelligence. The key here is to link the specific enzyme deficiency and the main GAGs that accumulate. Heparan sulfate and dermatan sulfate are common culprits to the clinical picture. For example, GHGs are critical for skeletal development, so their accumulation logically leads to bone and joint problems. They are also found in the cornea, so buildup causes clouding. Finally, let's discuss eye cell disease, which is like the master key failure for the lysosome. In this disease, the problem isn't one specific digestive enzyme. The problem is with the delivery system. Normally, lysosomal enzymes are tagged with a special sugar marker, mannose 6-phosphate, in the Golgi. This mannose 6-phosphate tag is like a zip code that tells the cell, send this protein to the lysosome. In eye cell disease, the enzyme that adds this tag is defective. As a result, all the powerful lysosomal enzymes don't get sent to the lysosome. Instead, they are mistakenly secreted out of the cell. The lysosomes are empty, and cellular waste products, including GAGs and glycolipids, build up. This results in symptoms that are a severe combination of the different MPS disorders. We touched on glycoproteins earlier, but let's really dig into the differences between the two main types, O-linked and N-linked. This is a classic exam topic. The names refer to the atom on the amino acid that the carbohydrate chain attaches to. In O-linked glycoproteins, the sugar chain is attached to the oxygen atom of a serine or threonine amino acid. In N-linked glycoproteins, the chain is attached to the nitrogen atom of an asparagine residue. This might seem like a small chemical detail, but it leads to some very big differences in how they are made and what they do. The synthesis process is a major point of contrast. As we mentioned, N-linked glycosylation is more complex. It starts in the rough ER with the construction of a large, preformed oligosaccharide on a lipid carrier called dolicol phosphate. This entire block of 14 sugars is then transferred all at once to the asparagine on the protein. This happens while the protein is still being synthesized. After this transfer, the sugar chain is extensively modified and trimmed as the glycoprotein moves through the ER and Golgi. This complex process allows for a lot of quality control and intricate signaling structures. O-linked glycosylation, on the other hand, is a simpler post-translational process that happens almost entirely in the Golgi apparatus. There's no dolicol phosphate carrier and no preformed block of sugars. Instead, the sugars are added one by one, directly to the serine or threonine on the completed protein. The chains are generally shorter and less branched than N-linked oligosaccharides. Think of it as custom detailing versus a prefabricated part. This type of glycosylation is common in proteins that form mucus, like mucins. The many sugar chains help these proteins attract water, giving mucus its protective, gel-like consistency. So what's the clinical relevance? N-linked glycoproteins are incredibly diverse and include most proteins secreted into the blood, like antibodies, immunoglobulins, hormones like TSH, and clotting factors. The complex N-linked chains are often crucial for proper protein folding, stability, and targeting. I-cell disease, with its faulty mannose-6-phosphate tag, is a disease of N-linked glycoprotein targeting. O-linked glycoproteins are key components of the extracellular matrix and mucus. For example, the ABO blood group antigens are O-linked oligosaccharides on the surface of red blood cells. The distinction is fundamental to cell biology and has direct implications for everything from immunology to endocrinology. Okay, we've covered a ton of ground. Now let's boil it down to the high-yield must-know points for your exams and give you some ways to make it stick. First, remember the locations. Protein synthesis starts in the rough ER, but the complex sugar additions and modifications happen mainly in the Golgi apparatus. The breakdown of JAGs and glycoproteins happens in the lysosome. This cellular geography is a frequent source of exam questions. Remember that the lysosome is the cell's recycling center, and when it fails, you get storage diseases. For the mucopolysaccharidoses, focus on the two big ones, Hurler MPS 1 and Hunter MPS 2. Memory aid: Hurler's hurts all over because it's the most severe, affecting skeleton, corneas, and brain. For Hunter syndrome, remember the mnemonic, Hunters see clearly, no corneal clouding, and aim for the X, meaning X-linked inheritance. This simple phrase helps you remember the two key distinguishing features of Hunter syndrome compared to Hurler. Also, don't forget I-cell disease is the shipping error. The mannose-6-phosphate tag is missing, so the lysosomal enzymes get secreted instead of delivered. When it comes to comparing O-linked and N-linked glycoproteins, think about complexity and location. N-linked is N-tricate and starts in the ER. A preformed block of sugars is added to asparagine, which also has an N in it. This is for complex proteins like antibodies and hormones. O-linked is simpler, happens in the Golgi, with sugars added one by one to serine or threonine. The O comes from the OH group. Think of O-linked for mucins and blood groups. Knowing where they are made and the amino acid they link to is crucial. Finally, my call to action for you is to not just memorize these facts, but to understand the principles. Draw out the pathways, explain them to a friend, connect the molecular defect to the patient's symptoms. Why does a gag buildup cause skeletal problems? Because gags are essential for cartilage structure. Why does it cause corneal clouding? Because the cornea needs to be transparent and cells full of undigested material block light. Making these connections is what separates simple memorization from true understanding. You've got this. Keep putting in the work, stay curious, and you will be an amazing physician. Now go crush that exam.